It's my great pleasure to welcome to the today extra talk Remco Soer, Professor Remco Soer, who is at the university, the Rijksuniversity of Groningen, UMCG, and he is well-established professor covering human movement science, focusing on pain. Originally educated physiotherapist, but by doing a lot of cohort studies in the real world, covering human movement science and especially pain, with thousands of subjects included. He made it to 75 peer-reviewed international journal articles, and collected a lot of experience in regard to pain design, or study design, including N-of-1 studies, which have tremendous relevance in the context of Selbstvermessung, so in English, quantify yourself. And we're glad to welcome Remco today in our group to give us an introduction covering the comparison of the randomized controlled trial, as Remco's title is presenting, which might be still the holy grail in medical research, and then the N-of-1 and maybe anything in between. So I'd like to welcome you, Remco. Thanks for joining us, and yeah, we're looking forward to your presentation.
Yeah, thanks, Sebastian. Very nice, and I feel very welcome here. So thank you for inviting me to this talk. Indeed, I would like to talk about randomized controlled trials and design. So it's going to be a bit of a methodological talk, but I think that for medical informatics, this is a very important topic as well. Let me start about introducing myself. So I'm an assistant professor at the University Medical Center in Groningen. I work at a pain center, and I'm an associate professor at Saxion University of Applied Sciences, which is located in Enschede. So I travel around from the north of the Netherlands to the east of the Netherlands almost every week. And I'm also the editor-in-chief of the Journal of Back and Musculoskeletal Rehabilitation. I live somewhere in between, at some of the highest mountains of the Netherlands, which are 50 meters high. So don't laugh too hard, but that's what we have in the Netherlands. I live there with my family that you see on the right, and that was a while ago when I turned to be 40. So I would like to talk about the randomized controlled trial, and to be honest, I did quite a lot of randomized controlled trials. When I started doing research, I thought really that the randomized controlled trial is just, it's the summum of original research. If you really want to have a high impact journal citation, and you really want to publish in The Lancet or Journal of American Medical Association or the New England, do the RCT and have a relevant topic, and you will be there. So we got this published in 2023. So this year, this was in the JAMA Neurology. So it's really top of the bill, and it is a randomized controlled trial. And for those not familiar with randomized controlled trials, basically what you want to do is you want to compare different groups to each other. And usually those groups are based on a different intervention. So what we did here, we wanted to compare whether surgery on neck herniation would be beneficial from the front instead of you do the surgery from behind. So what you do is, what we did was we included 389 patients that were all diagnosed with a cervical herniation, so a neck herniation, and they were all going to have surgery. And they had very strict inclusion criteria, so they had no comorbidities, they had no muscle spasms, they had no discopathies on other vertebral levels. No, they were 389 clean neck herniation patient who had the neck herniation on exactly the correct side, so that either a surgery from the front side or from the back side could have been performed. So we excluded some because people did not want to be randomized. They did not want to say, okay, I'm going to have a coin decide, or an envelope decide whether I'm going to be random, whether I'm going to have surgery from the front or the back side. Some did not meet the inclusion criteria because the neck herniation was a bit too much on the medial side, for example, and some just thought, well, okay, I don't want to participate at all. Then you have an eligible part of 265 patients, and you're going to randomize them just by the coin or the envelope in our case, whether you would have a posterior surgery or an anterior surgery group. So if you then have those two groups, you can completely compare the interventions together. And because you randomize them, you know that all factors like age or gender are randomized, and they would be fairly equal to each other. And that's what randomization is for. By randomization, you make sure that you will not get two completely different groups. It might turn in to be that, but if you have a good, well-established random group, you will have a good balance in both groups, basically all the time. Well, then you have a surgery, then you have a follow-up, and what you do is you compare the two groups together by statistical analysis and see who does best. You see these flow diagrams basically in all randomized controlled trials. They're very standardized. This is based on the consort statements for randomized controlled trials. And basically, this is just a way of good research. And if you look at the hierarchy of scientific evidence in original research, then you see here the randomized controlled trials are the highest, have the highest evidence. It's only been topped by systematic reviews, but that's basically not an original research, but just a literature review in which all the evidence based on randomized controlled trials, for example, are summed together. So it's not an original new study. But beyond that, the randomized controlled trial is on top of the evidence pyramid. And still, all the time, I thought, okay, if I see this patient as a physio, should I give the best treatment following the randomized controlled trials? Because my patient is a bit overweight, and my patient is not the same patient that was used in the randomized controlled trial because it has a double hernia. My patient is from a low socioeconomic status, and it lives in a difficult situation at home. So then the Dutch Council for Health and Society came out with a very influential report in the Netherlands, which is called Zonder context geen bewijs. Without a context, doing research in a clinical lab situation, basically is that what it is, you will have no proof, because you can see what happens in a lab situation, but when someone is getting out to work again, you do a herniation study like this, and people start going insane on their mountain bike, or they will be very hesitant to move. They'll have anxiety to move. So there's a big difference on how people will move afterwards. So basically, you don't know how they're going to recover in their own context, and that is very important if you do those studies. So the randomized controlled trial is a bit of an in vitro situation. It's a bit artificial. whereas functioning really represents a valuable action in a relevant context. So you really have to add that valuable context in order to be specific for a single patient. And if you look at the mean between two variables, you also see that within those groups, that it really changes, and there's a big difference between patient A and patient B without the group. So if we look at the validity, and especially the external validity, the generalizability of the RCT results to the general population, then you see a big inclusion bias. Basically, just the motivated patients will be included in your study, because you have to fill in a lot of questionnaires. You have to be willing to randomize yourself. And that takes quite a motivation. While usually those people that are not so motivated to change, for example, in their behavior, are also the patients that don't do so well. So you'll have an inclusion bias. And RCTs really don't like wide inclusions. What I said already, you need one single problem. So just the neck herniation, not on a different level. No two herniations, cannot be older than 65 years, cannot be any younger than 18 years of age. So you get a whole lot of restrictions, and basically you will leave up with a very small homogeneous sample, which has nothing to do with the real patient anymore. So I'm a really big fan of Trump, and basically everything which is a bit different than he is, it just out with him. So really, all the patients that are a bit different compared to normal are excluded from randomized controlled trials. If you look at the personalization of the treatment, RCTs are in common, they present averages. So the whole group is being averaged. Basically they don't say about how many people do get better, how many people do not get better. And if it is so, then still the question remains, who does get better and who does not get better? So who should we provide which treatment is a completely different study question. So the RCT uses the framework of, does a certain treatment work on a group level, but it does not give an answer on what works for whom. So it's great for policymakers, it's great for healthcare insurance, but it's not really for clinicians who have only that one patient and have to advise a certain treatment for this patient. So also one of the problems for randomized controlled trials is heterogeneity. There's a lot of individual characteristics in patients. Basically, they're numerous. You can randomize based on gender or age, but there's so many other characteristics that there are. For example, also in low back pain, the one has a herniation, as I said already, but the other one can suffer from post-cancer pain. So there's a lot of different subgroups assumed, for example, in low back pain, but we don't know exactly, based on what we should get, that heterogeneity is based on. So what we see is that single interventions frequently fail in a heterogeneous low back pain population, and that's also why we say, okay, if you just have one mark, just the surgery, should we also add rehabilitation, or should we also add anesthesiology? That all kinds of factors that are not really suitable for the randomized controlled trial. So if we take a look at it on the RCT, and just in a brief summary, the RCT basically fails in very heterogeneous groups. The randomized controlled trial it's very difficult to obtain adequate sample sizes. As you saw here, in my first example, we had 318, I think it was, subjects with a neck hernia, but we needed 4 years of inclusion, and we needed 10 hospitals, 8 or 10 hospitals, to obtain the adequate sample sizes. So they often ignore the individual patients, and they are in vitro representations. So I was a bit struggling with it. From the RCT, is this still the gold standard, or should we really do something else? So should we also get out with the RCT? I'm not sure, but at least it made me think, and it gave me. I went to the other side. I thought, okay, if this is not it, how about the complete other side, the single subject research methodology? Basically, a single subject is just, for example, an N of 1 trial, or a few N of 1 trial, or it's a case study. It might be an alternative to the randomized controlled trial paradigm, but then added with a tool to improve clinical evaluations. So the clinician might really benefit, or the individual patient might really benefit from a single subject research method, because that patient is not interested in whether a therapy works, yes or no. It just wants to be helped. And it wants to have the best treatment for him or her, not for the general public. So if we look at those single subject designs, what they have in common basically is that they have a baseline phase, they have an intervention, you could have repeated measures, you can control extraneous variables, and you can do analysis with them as well. So I want to go through those components and see what the strength of those single subject designs are compared to the randomized controlled trials. So if we look at the baseline phase, Sebastian, is it possible to have some interaction already?
Yes, of course, if you like.
Yeah, so all people can talk. So I'm just wondering, if you look here on the left bottom slide, what would you say if at phase 1 or day 1, you see that someone has a capacity to walk on the treadmill for 4 minutes. Then on day 3, so this is basically the intervention that you give. You give a training, and this is what you see. Would you say that this intervention would be successful, yes or no?
Assuming that a horizontal bar represents something like the standard of another intervention?
No, that's just the average.
Then to this degree it would work, considering the randomized control trial.
This is just an N is 1. This is one subject who is measured on 4 different days on a treadmill.
Then it's not working.
I think the question is what is the expectation? What should the person be able to do? Right? What is the normal situation, or what is he or she recovering? Because I mean, we don't have all this information, right?
No,
I mean, it might be.
I agree, but if you just look at the figure, would you say that here the treadmill in minutes, that they would increase, yes or no?
I would even say it has not a big effect. I mean, the first 3 look like, oh, it goes up, nice, right? But then, yeah, it's again. If you would then observe maybe the next 7 days, and it stays around this line, then you know it's more like this is the situation, right?
Yeah, it's a bit doubtful, right? From this, we can't really say that anything is happening in this intervention. So I would like to talk about the baseline phase, because basically we don't know. Yeah, correct, Martin. I saw your comment coming. This is basically the importance of setting a good baseline. So if this is the intervention, basically we don't know what to expect, because we don't know what the baseline would be. So I would really suggest in single case or in single subject designs to get a good baseline measurement with repeated measures. At least 3 measurements, but I would really prefer more than 5 before you will start with an intervention and really note it. And then you can look for stability or you can look for trends. But let me explain by this. So if you have your intervention and you have the same pattern, but then as a baseline measurement, and you will start your intervention at day 7, then you can really see something completely different, because basically what you see is that also the baseline is not a stable and a given situation. No, the baseline varies as well. And if you now see that the intervention at day 7 starts, then you still see a bit of the same of a pattern after the intervention, except for this is higher. So I don't think I have to ask you whether you think that the intervention would be suitable. Sebastian, you have an idea?
It should work, yes.
This looks like quite a good and an effective intervention then, right?
Yes, yes.
So there is, it's really important to have a good baseline. And you can look at trends, because those different baseline patterns will lead to different conclusions. So if you have a very stable baseline, then you know that the difference that you want to obtain during the intervention can just be very small. If you already have something like a learning effect, or someone is healing, or someone is deteriorating because it has an autoimmune disorder, which is just degenerative and it keeps on getting worse, or it fluctuates, what you see in chronic pain patients a lot, that the one day they have a pain score of 8 on a 0 to 10 scale, and the other day they say, okay, I'm doing quite fine, I only have a 4. So it fluctuates. Then it's really difficult to say whether an intervention would be indeed be effective, yes or no. So the baseline is perhaps even the most important thing in single subject designs. Get frequent measurements. And if you have, for example, wearables that you want to provide, first create a day-to-day baseline on how many steps you're having, or how reliable the thing works, or if the subject really wears the smartwatch, yes or no. Really important, get a good baseline. So that comes to the second phase, the intervention phase. What is really important is that based on the baseline, you will not change anything except for the intervention. So if you say, I'm going to train you for sports, or do a physiological training, then don't change anything in the behavior of the patient except for that training. So in this study from Pereira Gizzo in 2016, what they did is they first had a baseline in patients with cerebral palsy. So that is a brain disorder that children are born with. And basically what they did is they gave those patients, and there were just 2 patients, a digital memory game in which they could learn to remember whether something was a plate with cups on it, or something was a football, based on a card. And that digital memory game was after 4 games or 4 sessions. The 4th session, the intervention was provided, so the memory game was provided, and then you see that learning will occur. So on the first trials, you don't really see a difference, and which is quite stable compared to the baseline, but after a while, and that's what you would expect also, is that people start learning from it. So it did take a while for the child to learn, but it did do, and it went from 6 or 4 to 6 to 9, which is indeed an intervention, which is pretty effective. And I think that it's very important also to know that you keep doing the measurements at the same frequency as you did in the baseline to really be comparable between the intervention and the baseline. So that's basically what you want to do with an intervention. So the question is, what are you going to expect? Would this be an effect of the physiotherapy treatment? So if you look at the walking time, someone had a stroke, so they have been disabled. People start to do a baseline measurement on the walking time. And then the physiotherapy starts. So you have a quite stable baseline, and then this happens. So how would we interpret this? Anyone has an idea?
I mean, it looks like it's working, right? On the other side, of course, there might be effects where just certain things start to heal from a specific time on. And what you do as intervention only looks like it's supporting, but in reality, because you in the N1 trial, you don't have a comparison, and then of course you also wouldn't, because people are different, as we know, you wouldn't know if, let's say, the other one without a physiotherapy would have the same curve. Then you would say maybe, yeah, physiotherapy is not that effective because it would have come from alone. But as we know statistics, this is not really saying anything about the specific case, right?
Yeah.
And that's the topic. That's the problem, I guess, right?
Yeah, and I think you have a very important disadvantage or challenge that you have in such an A-B design. This is an A-B design, which has a baseline, an A, and an intervention, a B. An A-B design does not really rule out all the causalities that you can have on improvement. So indeed, it could be the physiotherapy that was effective, yes or no, but it could also be because after 6 days, the patient starts to heal by itself. So it doesn't say anything about natural healing or whether it's the physiotherapy, yes or no. And that's why you would, in a randomized controlled trial, you would need a good control group so that you could rule out physiotherapy versus no physiotherapy, for example, and see how the physiotherapy contributes on a group level. You cannot really see that here. So the question is, what would you expect? So how would you interpret your baseline and your intervention in A-B design? So if you look at the A, just short, effective or not effect?
We could do it on the chat, so everyone who is thinking that it's effective does a plus and the others do a minus, and then we average or take the mean. So there is more plus on A.
Plus, yeah.
Yes.
The second one, is that plus or minus? Also plus. Still effective? Yeah.
Plus, yeah.
C, is that effective, yes or no?
Plus, plus, minus, question mark. It depends, I would agree. Question mark is a good one. Yeah. A lot question marks.
Why would it depend? Who had a question mark? Is someone able to
So I had a question mark. I could start and then the others could join, because in the baseline you have a lot more fluctuation, and it's depending fluctuation is better than stability.
You don't know whether it is, I didn't say whether it should be better or worse, Sebastian. Basically, something is happening clearly, right?
That's a good one. Yeah.
So,
so will you redo it, everyone? Is something happening or not?
I think that makes it a plus, Sebastian. Yeah, I think so too, Andreas, that definitely something is happening. So this could, for example, be an ADHD trial in which someone fluctuates very much in things and becomes more stable in his behavior. So if you look at the right side, plus or minus?
Plus. Majority is plus.
Yeah?
Yeah.
Why?
Because it works later, but it has an effect.
Yes, but basically what we said in the last slide, that it was pretty ambiguous. We don't really know whether the intervention would work, yes or no. Yes, that is exactly it, Martin. It seems to have an effect. But we're not quite sure, right? Because of those two lows. How about B?
There we go with minus. Hard to say.
Yeah. Yeah. And C?
Minus.
Yeah, so basically what you see on the left side is that you really see a clear cut between the baseline and the intervention. But this is more ambiguous, and it's just hard to tell. It might be an effect. It looks a bit, if you look at B, it looks a bit higher, and it keeps continuing. That's beautiful, but you saw that same trend already in the baseline. That is exactly why the baseline is so important, to see what is happening. Otherwise, you would just say, okay, people start getting better. But what about natural healing? This would be much better. A would be much better, for example, if this patient is constant already over the years, and you don't know, and you just know that it hasn't become better for over the last 2 years, and then all of a sudden, if you become better, then you would say, indeed, okay, the chance is big that this would not be due to natural healing. But it's difficult, and that's why the interpretation of a single subject design is not always easy to do. So then it's important to have repeated measures, to have basically in a single subject design, the more repeated measures you have, the better you have it. This is from my Dutch colleagues that was on a body weight supported treadmill in patients with a partial spinal cord injury. And basically what they did is they had three different subjects, and they did a body weighted support treadmill training. And they have a baseline, and here they have the intervention. So the squares are the baseline, the triangles are the intervention, and the circles are the follow-up, and there's one late follow-up. And there they said, okay, this subject performance so on a quality of life index over time, this one on the Canadian occupational performance measure on performance, so how are you able to perform in your daily activities, on satisfaction, and on walking capacity. So you have a lot of measurements also on baseline. And then you see, for example, that body weight supported treadmill, it's not going to work in walking capacity in this subject. Not even for a bit. It is very stable. So you can you can just perform normal statistics on this, and then you would indeed see that this is a non-significant difference within that person on walking capability. So a body weighted supported treadmill training is not going to help, I think, this patient that much, unless you see others. Not so much in quality of life. It fluctuates. It's not so, a bit more above average compared to below, but the baseline average. You don't really see it in performance. While subject 2 really responded on the performance. You saw a baseline, and from intervention 1, this is the first one, you really saw a change in the self-report of the performance of the patient. So the patient felt that this body weight supported treadmill training would really give him the ability to walk again. And it really looks like people are going to walk again. I'm not sure whether you know these body weight supported trainings. So basically, it reduces your weight on a treadmill. So people with a spinal cord injury that have muscle fatigue and weakness are not able to support themselves on their legs. And then they come and walk in those supported treadmills. And then you can indeed train your muscles in walking again with limited weight. So this person really felt, I'm walking again. You can really see it. And you could also see that this patient was very satisfied with it because it was really opening his eyes. And basically, if you look at the walking capability scale, you could also see basically an immediate effect. And the most beautiful thing was here that it did not stop. So when the intervention stopped in the follow-up, this patient reported that his walking capability was still quite high. While here you might see some washout. So the satisfaction decreases a little bit. You do the intervention and they were really hoping for it, that their walking capability became better. But then when the intervention stopped and people went home again, they had to support their whole body again. And they said, okay, well, this was a bit of an in vitro situation. And you also didn't see a lot of those things in quality of life. So what works for the one subject might not have been beneficial for the other one. So in subject 2, they might have had a real benefit, but not for subject 1. If you put those in a randomized controlled trial, then on an average, probably you would have seen a slight effect. So it works on an average, but it did not work for subject 1, while it did work for subject 2. And this is definitely important for an NS1 trial and for a clinical application.
Yeah, Remco, how would you, I mean, theoretically, if you would do a randomized controlled trial, or let's say, or compare different groups, however, right? And then, but let's say all of this data would be captured. Could you then say that also from the clinical trial you could draw some conclusions? Because sometimes, I mean, when I think about let's say research in cancer or in other areas, right, where there's always the idea to find out some biomarkers to why it works in some of the population, in some parts where in the clinical trial, and for some it doesn't work, then it always like is indicating there must be a biomarker or something like that, or there's a hope to find some where you can say, okay, it doesn't work for everybody, but in case you have this mutation, for example, then it works quite well for you, right? So
Yeah.
that's we all know about that part of the business, right? And the question is, in let's say other areas where it's not about let's say drugs, or where it's really about these treatments, right? Is there also a try to get research done to find out, or maybe which subpopulations, even based on a multiple of these n equals 1 trials, can help, and for which others maybe another intervention must be considered, right? Because I mean, people want to get help, and the doctors also,
Oh, yeah.
physiotherapist wants to
Yeah.
help them, right? But then, of course, it's the game to find out what does really help and what maybe is just a waste, right?
Absolutely. And in randomized control trials, they also want to look at different subgroups. So, for example, they can see whether the intervention might be more suitable for men versus women. But there's just so many factors usually that that vary within a group. So it's not just it works better for men than for women. It might also work for men who for people who are less anxious compared to others. So, for example, that subject 1, he might be a bit afraid to walk on the treadmill, so he would not be satisfied. Or he might have been very old and he didn't want to walk anyway. Or so it's or it doesn't work for that group. And in randomized controlled trials, if you do those subgroup analysis, you very quickly have a problem with your power analysis. So you need, actually, if you want to do those subgroup analysis, you might want to double your population. So you need higher numbers to really state something about it. If you want to do it for really for an individual patient, then we come to another design, which you can also do for a single subject design. So I'll get to that in a few minutes. Okay. What I wanted to add is that if you have a lot of measures, you can also just do your baseline, distract your baseline from your intervention, or your intervention from your follow-up, and then you can just do kind of normal statistics, repeated measure statistics also, to see whether this is a significant difference in those repeated measures. So that's really an advantage as well. Then there's a lot of these designs. We now only talked about the A-B design. So you have a baseline and an intervention, but you also have a baseline, intervention, baseline again. So if you want to see whether this was due to natural healing, yes or no, then do another baseline. Stop the intervention and see what's happening. If it returns immediately to the old situation, then probably it was not due to natural healing. So it gives you an additional idea whether the true effect was due to the intervention, yes or no. You can also do it again. You can do a baseline, intervention, baseline, and do an intervention again. If it's similar, the patterns are similar, then indeed the chance that this intervention really works or does not work, do the opposite, but indeed does something, is absolutely getting higher. What you can also do is multiple baselines. You can also do an ABAC, and this has been performed quite often. An ABAC basically is you have a baseline, you have the intervention, which works very well in medication trials, for example. You see what is happening. You'll have another baseline, and then you do the intervention, except for you will use a sham. You will have a placebo, a pill, which doesn't work. And if indeed it will remain stable over time and it does not change from baseline, when you do the sham, then the chances that the true intervention really works is much bigger. So this is your own control group then. So you will have a placebo-controlled study in an NS1. And I think that this is very beautiful. And you can also do an ABAC and a BC. So the C does not have to be a sham, but for example, if you say, okay, but I want the combination of two interventions. I want rehabilitation and surgery combined and see what that happens. Or I'll give two pills, morphine and paracetamol, and see what that does together on pain experience. So you can also do the interactions. So there's a lot of designs that you can really do for an individual patient to see what would be the best. Should I give this patient morphine for the pain, or should I just give them paracetamol? So you give an A, B, A, C, or should I give them morphine and paracetamol? B, C. You can really check it out. And there are clinicians that really do those things. And see whether for this patient, this medication helps, yes or no. So the question is the N of 1 trial a worthy alternative? And the N of 1 trial is something different than a case study. It's also not an N is 1 trial, because the N is 1 trial is just the trial that you have on the single case, so the single subject case. The N of 1 trial basically uses one participant, and I'm not sure whether you can see this, but this is the traditional trial, and here you need a lot of participants. The trial duration is very short. You'll have an individualized treatment plan. Data analysis are rather simple. It is patient-centered, and basically it's quite cheap. So a patient does not have to wait for the results for years, but you can just experiment with your own patient. And the N of 1 study is a trial. It's a design in which a single patient is the sole unit of the analysis, and it acts as his or her own control. So they usually receive a different treatment option or a placebo, so that would be the ABAC design, but you do need a washout period. Because when learning, for example, is involved, then you don't see that washout anymore. And then you don't see the difference between the intervention and the baseline that follows, or the intervention that is afterwards, because people have already learned or changed their behavior. And then it really allows for the intraperson comparison. And you can definitely also randomize them. So you can even blind yourself. You can blind the patient on what intervention you're going to provide. So if you have the pill, you have basically a double-blind, placebo-controlled trial, because you give the pill that is in the one envelope, and of course you need to know what it is after a while, and that might be the placebo pill, and then afterwards you give the real pill, and you just follow the differences of the patient that are there. That really is a very high quality evidence in medicine for the N is 1. So that is also why the N of 1 randomized controlled trial is considered a level 1 evidence also. And I think it deserves this title as well, because I think that it is very important that for the individual patient, it's just the matter how you focus. Do you focus on a big group, but not on the individual patient? Then for healthcare insurers, this is very valuable. But for the clinician, if you do an N of 1 randomized controlled trial, I think that this is much more valuable for the individual patient. So you'll have the different ABA designs. but really the randomization and also the blinding that you can do in an N of 1 randomized controlled trial is very important to get that level 1 evidence. So I hope that many more N of 1 trials will be published, because you don't see a lot of them in the literature as of yet, but they do gain popularity. So the N of 1 trial within patient randomized blinded placebo-controlled crossover comparison, you can really do them in medication study because you can really blind them. They have a very high value for patients. And again, you can see the baseline and the differences. You can look at differences in trends, but you can also do some statistics on it. So what of course you want to do is to quantify the impact of your intervention. So you can do it a bit like eyeballing. You have a baseline, which is the same baseline as we had in the beginning. Then you have an average of an outcome of 5. Then you see the difference after the intervention, which has an average of 10. So the impact might be somewhere around 5. Whether that's clinically relevant, yes or no, we don't know. But you can do it with eyeballing. You can really see that basically this should be effective. And another way that you can use is you can use the 2 standard deviations that there are based on the average. So this would be the 2 standard deviation band width, in which 95% of in a normal curve, 95% of all values would be. And basically you can indicate statistical significance, and what literature says that if you have few, 2 or a bit more, it depends a bit on how many measures you have, for example, that you have outside the 2 standard deviation band, that may indicate a statistical significance as well. It does not tell you something about the clinical relevance. So you really have to quantify that impact as well. What would you state would be valuable to the patient, yes or no? So there are some challenges, but there's also definitely some benefits on single subject designs. So what you do see is that they have limited generalizability. What works for that one person is not necessarily to happen or to work for the other person as well. So you can't see it like it would help for everyone. There's potential confounding. What one of you said, I think it was Andreas. It might be natural healing in an ABA design, but an AB design, but it might also be that other confounders are there. I broke a leg, for example, and then it didn't seem to work at all. Especially in an N of 1 or an N is 1, that has huge impact. The statistical assumptions may be breached, so you might have too little data points to really perform statistics. But on the other side, the benefits are that you'll have a great individual effects. You can apply it in very many settings. If you are just inventive and creative and look at a good design, you can basically use it for every disorder and every intervention. You'll be quick. You need very small numbers. You'll be cheap, and you'll have a very high impact for the one patient. And it has a level 1 evidence. So I'm really fan of the N of 1 controlled trial. So I hope to get more of those chances to do that. What we do see at this moment is that we follow large cohorts. And based on those cohorts, and especially newer techniques, I do this, for example, together with Sebastian as well, because he's the machine learning expert here. This is a Bayesian network that we made, and basically we were also looking at what works for whom. So what kind of characteristics make that the one patient with low back pain has a positive response of surgery of multidisciplinary rehabilitation or anesthesiology. So if, for example, something is really wrong in your back, probably you would need surgery. Quite obvious, right? So if we have data from a few thousand people, then a machine learning algorithm might be able to capture those patterns and see which patient with what characteristics needs what treatment. So what we did is we put all those data and follow-up data in the model, and what you do see is that the red lines, the red arrows, are the significant arrows, and what you see is that having irrational thoughts, stress, being extremely nervous, so those psychosocial factors are really related to the duration of pain. But that's basically not what we wanted to know. We wanted to know, should we provide them with rehabilitation, surgery, or injections? And you see that they're all black lines. So there's no significance, and those machine learning algorithms still cannot predict what kind of treatment someone has, and also not what some of the treatment responses would be. So whether there has been a trauma and whether that person has a serious disease based on that trauma or should get surgery based on that trauma, we don't know at this moment. So basically, we're still a bit in the dark here, and we don't know, based on large data, whether someone would be beneficial to a surgery or a rehabilitation or whatever. But perhaps you could do this in a good N-of-1 design. It brings me a new idea, Sebastian, what we could do. Perhaps we can even combine the two, but that's for later. So the take-home messages is that the randomized controlled trial is not always a suitable design. It's expensive, it's time consuming. I think it takes about 400,000 euros to do a really good randomized controlled trials. You need large samples, you need a lot of time, you need a lot of patience, and you will get an effect based on groups, not on individuals. And the single subject designs might, on the contrary, be very worthwhile for the individual, for the clinician, for personal cases for patients in their home context. So basically the take-home message is that I don't think that there's a real good or bad here. We definitely need randomized controlled trials because we want to know whether the healthcare insurance will pay for the one treatment, yes or no. However, if you want to know whether the one patient really benefits from his or her therapy, then you need the N-of-1 trial. So a good method would really fit the research question. I think that's the most important thing that I want to deliver for you today. So thank you very much, and well, basically the floor is open for questions or discussion.
Thank you, Lord Remco, for this wonderful enlightening talk. I think you made it really clear what's the differentiation between randomized controlled trials and N-of-1 trials. And you made a good point also in determining special cases, clarifying which conditions to be considered especially. Yeah, let's open for questions in case there are already some of the auditorium. I'm looking forward to this. Meanwhile, I might have one question. So when we are considering in this bigger XtraTrack panel, on the one hand, the clinical side, like we had a wonderful talk of rare diseases, where I think N-of-1 trials, we don't have to talk about it. As you motivated it, we have limited...
You cannot sufficiently power them. So especially in rare diseases, I think N-of-1 trials are very highly valuable. Yeah.
Yes. Yeah, and then, but on the other side, like, so you gave yourself a good other clinical use case that's reasonable. But on the other side, we are also discussing this fitness, self-tracking, quantify yourself users, which are aside of the health part. I mean, they want to stay longer healthy, and we covered in the last presentation some of the motivational aspects as well. But what about them? So there, it might be much more challenging to have randomization, to blind in a way. So do you have a hint for them? What would be a good approach in this direction to do an N-of-1 trial? And in that regard, also, do you have a feeling, is there something to quantify the reliability of the design I'm using N-of-1, like AB, ABA, and then as well the degree of blinding, randomization, treatment period, considering washouts. So how could I determine how reliable my trial design is?
That's a very good question. Basically, evidence-based medicine is a combination of good clinical practice. So you need experience. You do need to know a lot of the disease characteristics before you say, okay, you definitely need to know how long your washout period should be, for example. So you need a lot to know on pharmaceutics. So you need disease-specific insight. You need a methodology. But if you refer, for example, to wearables, they might be difficult to blind. However, I think there is also quite some wearables that you can blind or program. I don't know, as a placebo. You just say on your watch, you can program on a small app, perhaps, that someone is always behaving like they did better, and you give them like a fake motivation or something like it.
And I mean,
you could
even, for example, design two fitness programs. The one having an effect, let's assume, and the other one, it's rather cardio in a low degree, so wouldn't have that big effect.
Yeah, but probably you might, you can blind the patient with it, but it might be difficult to blind yourself as a professional then, because you might just know what you do and what you provide. And usually the professional can see very quickly on, okay, this is the placebo that I'm providing, yes or no. Yeah, but there's
so you would agree
capabilities.
Yeah, so you would agree that that's one of the challenges when doing it on your own.
Yeah, I would say so. Yeah, but perhaps if you have indeed a student or an assistant that can help you with it, then you can blind yourself as the data analysis or analyst. So then in some way you still have some kind of a blinding. However, the therapist might not be blinded and might therefore also be something like a placebo or a no-semi.
And there might be a big benefit or potential for in the next talks we will discuss, like regarding general health data platforms, European health data space in particular, and we will have a company which designs circular business models around health data, and I think that might be a benefit for them to support users in the fitness domain by designing some of these trials for them and then having them give it a try on their own.
Yeah, in general, those big initiatives, it's always the question, how valuable is the information that is in there? For the, for example, you might see how the average German person moves, but you don't know exactly what's happening, right? So it's difficult to find effects in there because you miss so many data as well.
And the only benefit could be on the individual level that aggregating multiple data sources into one data set, you have better view on the individual, as long as you don't anonymize. Yeah.
I'm sorry, you got stuck a bit, so I couldn't hear you quite.
Okay, and the benefit of this bigger European health data space, not but maybe electronic patient record, might be that you combine the data of multiple sources, of multiple sensors, entities, and
I think that this really is the future, Sebastian. There's so much data that is being gathered just in regular care, but it's not always structured, and we don't know exactly. We have to report a whole lot of data as nurses and doctors and physiotherapists and healthcare professionals in general. But we never do anything with it, and I think there's very valuable information in there. So if we can combine this with qualitative data, then and I think that we will point that out, Sebastian, right?
Yes,
That's what we're
cite
exploring.
you in some days. So another question, and there was one of Leah. Do you want to ask on your own? Or shall I ask?
I could ask. It's just about bias and confounders. Do you think the confounders are easier to identify in one and like N is one studies, or is it easier to exclude the confounders because of the large numbers of cases in an RCT?
Yeah, that's a great question. Basically, what I said, it is very important that the only thing that you try to change in an N of 1 design is indeed the intervention, so that you will have as little confounders as possible. But of course, someone can break a leg and things turn out to be bad. Well, this was not intended at all, and I'm using the break your leg because this has a serious effect, for example, on your walking capacity. On the other hand, if you have a good carried out NS1 design, then probably if someone has broken his leg, then if you do another baseline, you will see no changes as well. So or if you use another intervention like a placebo intervention, then you're still working with that confounder, right? So you will be able to identify that confounder, and probably if you have a very big group in a randomized controlled trial, confounding is ruled out based on group size. So based on the randomization and based on the large groups, probably all the confounders that there are equally distributed among both groups. So it's a different way of controlling for confounders. But I'm not sure whether the one or the other would be better, yes or no.
So in a, sorry, don't want to.
Now, what you usually do is in some of your first tables in a randomized controlled trial is that indeed you test whether the randomization was successful, yes or no, and whether age or other factors indeed were equal between the groups.
So in a N of 1 design, you need a close relationship to your patients, I think, because you need to know a lot about them and the context, so that you can find these confounders.
And you definitely need a good bond with, and because you need a lot of measurements. While it is not so time consuming for a researcher, it is quite time consuming for a patient.
Okay.
Because you have very many repeated measures. So people have to come back, or they have to really register at home what they've been doing, yes or no. So for a good within measure, But you can control for confounders as well in those NS1 trials. Yeah.
Okay, thanks for the answer.
Yeah.
And that's a real great question. And adding to it, so shedding some light on the dark side of N of 1 trials. Like you said, one disadvantage of RCTs would be that rather stable age group is included. And we could ask on the same side, like, is an N of 1 suitable for multimorbidity and older patients, or do we have so many confounding effects included? And on the other side, so another aspect in the same direction would be, like you said, as a disadvantage of the randomized controlled trials, that you need people who are really willing to do it. But as you said just recently, you need that maybe as one for the N of 1 trials, because you need to follow through. So there's no benefit if the people drop out after 2 months or so, 1 month or something. What do you think about that?
I agree, Sebastian. And basically what it is, you enter an RCT almost not for yourself, because you don't know whether it's going to be beneficial for you, yes or no. You might just as well be in the placebo group and never get a treatment, yes or no. Basically, you do it for the ones coming after you, you know, for the development of a new treatment. The N of 1 trial, you really do for yourself. You're really going to find out what works for you. And you can do it very well, especially with multimorbidities and in which a doctor, for example, doesn't know how the interaction effects are with your different morbidities, what works for you in this difficult situation. You could never enter a trial, but you could enter this N of 1.
Yeah, I take the first argument, so it's a good point, one of the best for motivation to keep the people staying with it.
Yes, absolutely. If they have a question that they want to be helped in the best possible way, then do something like this. Yeah.
Yes. Yeah. And for the second, I was thinking vice versa. So if I have multimorbilities and the patient's performance is varying on a regular basis among this N of 1 trial, wouldn't that be a challenge? So then you would have to have multiple repeated sessions of the intervention to cover this in a way, right?
Yeah, I think so too. Yeah, but I think if you are creative and you know something about what to expect, like washout periods, like learning effects, and you can just do a pilot with it, just as long as that you have the effect that you want. But I know that doctors and clinicians are really struggling with those N of 1 trials, and how exactly do I do this, you know? And I think if you cooperate as data scientists or as methodologists and the clinicians, that you can be very valuable to have impact for the individual. Yeah.
So thanks a lot. I'm looking to the others. Are there any further questions? Okay, in that case, I have one last question. So we have to focus on minimizing the unintended variation among measures in N of 1 to assure the meaningfulness. But this might consider or take effort and might limit the follow-through rate. Do you have any further solutions? So we figured out that the motivation is on the one hand because you do it for yourself, for your own health. That's a good, strong motivation. But regarding follow-through, is there any benefit? Because I assume you can't give a feedback in between because that would affect the trial, right?
Yeah, but on the other hand, if you want to change something in between, you can just do it in an interaction design, and you can just add another baseline and then do the interaction, or then do the intended change. It's not a method that has to be completely. You can vary your method based on, okay, this is what I want to do now. You can even do like a stepped care approach in an N-of-1 trial. So it's not that you have to set the exact amount of treatments, know when something is effective or not effective. You can even change your outcome parameters if you have them in your input. So you can really vary about it. It's much more up to your own imagination, as long as you just keep up with the methodological consequences and you follow the rules of an N-of-1 design.
Mm-hmm. And maybe a last question, so looking to the clinical use. So far, I see N-of-1 mainly being used in the research context. Do you assume once we're there that medical professionals are using it directly with patients? Would they do it on their own, or do they need some systems which support them in doing it?
Yeah, that's
communication, data analytics, keeping the data clean, the contact with the patients. There is a lot of efforts involved, right?
A trial is research, Sebastian, I think. So an N-of-1 trial will remain research. But basically, is this not what clinicians do every day? A bit slobby, I admit. But they use the one medication and then they say, okay, how was this? And then they use the other medication. How was this? And they don't know exactly what is happening with their patient. So basically what they do is they have a good idea, but they don't know really how to standardize it, and they don't know how to exclude themselves as an important factor in the trial as well. So they do it half, and I think if you really try to standardize those things, this is the search that doctors do every day.
Mm-hmm. So not replace gut feelings by centralized health platforms which could standardize processes.
I think if you standardize this, then you would get much more valuable information. Yeah, and you'll have a great N-of-1 trial to publish about as well.
Excellent.
That's what clinicians don't want to do as well, Sebastian. That's up to you and me.
I totally agree. But that's an excellent question for the next presentation, which will be next week, I think. Where we move over to Health X, so having a look to the European Health Data Space and data integration and data usage. For now, Remco, as there are no more questions, let me thank you. You really made it, so that's a wonderful presentation, a huge contribution to the XtraTrack series. And yeah, let me thank you, and hopefully see you very soon in person.
Yes, thank you very much, and thank you all for listening and attending, and I hope you can benefit on it.